Axolotls can regenerate their limbs, but how? Well, we finally have an answer to that question because scientists just discovered exactly what genes are activated when an axolotl regenerates a limb. And it turns out that these little salamanders may have a lot more in common with us than previously thought. The axolotl has long been famous for its crazy regenerative abilities. These little guys can regrow entire limbs if they lose them. They can even fully regrow sections of their brains and spinal cords. But until recently, we didn't know how the axolotl's genetic makeup allows for this extreme form of healing. In 2023, researchers from the University of Kentucky published a paper detailing how axolotl cells use M6A modifications to regulate gene expression during the early stages of limb regeneration. In other words, they figured out which genes need to be activated for an axolotl to regrow a limb. To do this, they performed several experiments involving amputating the limbs of multiple axolotls. Then they isolated the cells responsible for regeneration at various points after the amputation. To determine which genes were active at each point in the regeneration process, they used a technique called RNA sequencing. This method looks at the sequences of all the messenger RNA molecules in a sample. Messenger RNA or mRNA is like a template that tells the cell how to make proteins. It's basically the instructions. Proteins are what actually carry out the functions in cells. So by looking at which genes are being expressed as mRNA, we can figure out what processes are occurring in a cell. After amputating the axolotl's limb, the researchers gave the salamanders time to heal. They then isolated the cells near the wound at 4 different time points during the first week of regeneration. They chose these time points based on key events that occurred during the process. At 1 day post-amputation, cells begin migrating to the site of the injury, and by 3 days post-amputation, they start forming a blastema. If you don't know what a blastema is, it's essentially a mass of cells that's similar to a limb bud, which is the structure that gives rise to limbs during embryonic development. The cells within the blastema are de-differentiated, meaning they go back to a stem cell-like state and become capable of forming many different kinds of tissues. From there, the cells in the blastema begin to differentiate again, eventually forming a new limb. The researchers analyzed samples from these 4 time points to identify which genes were active during each stage of the process. They found that at the earliest time point, 1 day after amputation, a group of genes known as immediate early genes or IEGs were activated. IEGs are usually involved in cellular stress responses. So it seems that the cellular stress caused by the amputation triggers these genes to turn on. What's interesting is that the set of IEGs activated in the axolotl limb is very similar to the set of IEGs activated in human neurons following brain injury. The researchers say this suggests that axolotls could be a good model organism for studying human regeneration. They also identified some other interesting genes that were highly expressed at this early stage, such as a gene called IGF-BP5. In mammals, this gene is only expressed during development, but in the axolotl, it's expressed in adult tissues, and its expression is turned up during limb regeneration. Now, this doesn't mean that the axolotl has a special developmental program that it turns on when it regenerates a limb. Instead, it might have co-opted some developmental genes and started using them for a new purpose. Co-opting existing genes for new uses is a common theme in evolution. For example, feathers likely evolved when a group of ancestral birds appropriated genes that were previously used for color patterns and put them to use in building a feather. So these genes were not created anew, but were instead repurposed. Moving on to the later stages of regeneration, the researchers found that a gene called FABP7 was highly expressed in the blastema cells. This gene helps regulate the WNT signaling pathway. If you've heard me talk about regeneration before, then you probably already know that the WN pathway is really important for this process. Lots of studies have shown that turning up the activity of the WNT pathway promotes regeneration. Meanwhile, turning it down inhibits regeneration. So it seems that FABP7 is helping to activate the WNT pathway and promote regeneration. Interestingly, a mutation in this gene causes skeletal defects in mice. So it appears that FABP7 has a conserved role in regulating cell differentiation across species. The team also found that another gene, LMX1B, is highly expressed in the blastema. and this gene is important for patterning the developing limb. That is, it helps determine where different parts of the limb will develop along its axis. Now, it's not the only gene involved in this process, but it does appear to be a major player. For example, in chickens, LMX1B is needed to create the limb bud. And in humans, mutations in this gene cause ham-foot genital syndrome, which is characterized by underdeveloped hands and feet. Finally, the researchers identified a gene called CDK11B that was highly expressed in the late stages of regeneration when the new limb is almost fully formed. CDK11B belongs to a group of genes that are associated with cell cycle regulation. Even though the cells in the blastema are actively dividing, they don't have the typical characteristics of dividing cells. For example, they don't contain centrosomes, which are structures that help separate DNA during cell division. And they don't have a lot of cyclins, which are proteins that help regulate the cell cycle. The discovery that a gene associated with cell cycle regulation is highly expressed during the later stages of regeneration is quite surprising. The researchers suggest that this gene might be required to help reprogram the cells to allow them to divide once more, returning them to a more typical cell cycle. The study revealed a lot of details about the genetic basis of regeneration in axolotls, but there are still plenty of questions left to answer. The team hopes that future studies can build on this research and identify the molecular mechanisms that control these genes. Ultimately, understanding the molecular basis of regeneration in axolotls could help us find ways to stimulate regeneration in humans. For example, researchers are working to harness the power of the immune system to promote regeneration. Scientists at the Stowers Institute for Medical Research in Kansas City, Missouri, have made some exciting discoveries in this area. In a 2023 paper, they showed that after a mouse loses a toe, immune cells migrate to the site of the injury. When the mice were genetically engineered to lack certain immune cells, they regenerated their toes more quickly. The scientists think that this is because the immune cells release signals that interfere with the regenerative process. By better understanding the role of the immune system in regeneration, we might be able to find ways to reduce inflammation and accelerate healing. This is a very promising line of research, but it's going to take time. We may never be able to grow new toes or fingers, but at least we now know which genes need to be activated to regrow a finger. And who knows, maybe one day we will be able to regrow whole limbs. And while you wait for that technology to be developed, you can learn about the amazing animals that inspire it. The regeneration research I talked about in this video was inspired by axolotls, but axolotls aren't the only animals that can regenerate their limbs. Check out this playlist for more incredible examples. Thanks for watching and learning with us.